All right, so this is Unit 4 review session. Unit 4 looked all at enzymes. And enzymes are something that is really difficult for some students to understand at first, but if you just stick at it and stick with it and keep trying to understand this topic, you'll realize it's not quite as difficult as you thought it was. Enzymes are simply just types of proteins in your body that help to speed up chemical reactions in your body. If you didn't have these enzymes, well, then your body wouldn't be able to do anything. We need these enzymes to speed up all those chemical reactions happening inside. So the first objective says describe enzyme action in terms of enzyme substrate interactions and activation energy. Well, as you can see just from this first picture here, is that the enzyme here interacts directly with this molecule called the substrate. The substrate is just a fancy name for the reactant. So this is what you start with in a chemical reaction. So it's what is changed in the chemical reaction. So the enzyme interacts with this molecule and helps to change it. And the place where it interacts with it at is called the active site. By interacting with the reactant, or in other words, the substrate, the enzyme can speed up the reaction. And one thing I want to show you here is its effect on what's called the activation energy. So here you can see a graph that shows the progress of a reaction. This is without an enzyme. And it tells you the amount of energy in the reactants. This is what you're starting with. And the amount of energy in the products. However, you can see that there has to be this large input of energy for the reaction to start. And we call this the activation energy. This is the energy required to start a chemical reaction. However, when an enzyme is present, it lowers the activation energy. So now the reactants can somewhat be lazy. They don't have to climb that huge hill anymore. Now the activation energy is much lower and the reaction can progress faster. So let's look a little bit more at how enzymes actually do this. So as I said before, what happens is the enzyme binds directly with the substrate. So this is the thing that's going to be changed in the chemical reaction, the thing in green here. So it's actually going to go into this location called the active site. This is where the reaction happens, the change happens. The substrate fits perfectly into the enzyme, creating this enzyme substrate complex. In reality, the enzyme actually changes its shape slightly to help hold that substrate in place. And then the change happens. The substrate's turned into something new. And we call that new thing the product. And then the product leaves the active site. The enzyme is not changed at all in this chemical reaction. So now it's available for another substrate to come in and bind to it. And the cycle can just continue over and over again. So here you can see an example. This is an enzyme called catalase, and it breaks down hydrogen peroxide into water and oxygen. Hydrogen peroxide is a thing that is being changed, so hydrogen peroxide is the substrate. Our products are water and oxygen. Notice the enzyme is not being altered at all. So hydrogen peroxide comes in, binds to this active site. Here a water molecule leaves. That's one of our products. But you can see that we need two hydrogen peroxide molecules in this case to create both water and oxygen. Okay, but the enzyme can keep on working over and over again, so another substrate can come in and it can be changed. That's all that's happening. Hydrogen peroxide is coming in. It's being changed again into water and oxygen. Okay? So all that was happening there was hydrogen peroxide turned into something else. It turned into water and oxygen. Hydrogen peroxide is a thing that is changing, so it is a substrate. Catalase is a thing that's helping it change, so it is the enzyme. And water and oxygen are the products. So enzymes can actually break things down. As you can see in the middle here, this molecule is coming in. and it's being split into two different things. Here you can see that nothing's being broken down or built up, but A is coming in, and the atoms in this molecule are just being changed or moved around, and something new comes out, a new product. But notice the reactant, or in other words, the substrate, is a thing that is changing. Each time it's turning into something new. You can also have two substrates, C and D here, coming into the active site. They're actually being put together in this case, and they are turning into something new, which is letter A here. Okay, so enzymes do all this work. They cause change to happen. Another word for an enzyme is a catalyst. So a catalyst and an enzyme are the exact same thing. A catalyst is anything that can cause change. A person could be a catalyst if they evoke change. And an enzyme is a special type of catalyst. The next objective says, describe the effects of temperature and pH on rates of enzymatic reactions. So just remember that the relationship between temperature and the rate of an enzymatic reaction, or the rate of enzyme activity, will always show the same relationship. It's going to increase, the rate's going to increase to a certain temperature. We call that the optimum temperature. And then the rate is going to decrease. So why do we always see this bell-shaped curve? No matter what, you will always see a curve like this, where it goes up and then down. And this is when we're talking about the effect of temperature. Well, the reason that it's increasing here is the molecules are moving faster. So the substrate and the enzyme are coming together more often because they're moving faster. So there's more of a chance for that substrate to enter the active site. However, when temperatures get too hot, then the enzyme starts to fall apart. And when it starts to fall apart or lose its shape, we call that denaturation. Know this word. This is a very important word. If denaturation occurs, then an enzyme loses its shape and it can no longer function. Another thing that can cause denaturation is extreme pH. So notice we have the exact same shape curve here. So there's always some optimum pH where the enzyme works the best, but anything lower or higher than that causes denaturation. The enzyme can no longer function. It can no longer bind with the substrate and cause a change to happen. The next objective says recognize the importance of cofactors in enzymatic reactions. Cofactors are an ion or metal, such as iron, zinc, and copper. These normally occur or present in your body, and some enzymes require these. So these are cofactors for those enzymes. If they're not present, then the enzyme can't function. Not every enzyme has a cofactor, but many do. So if you remove iron or remove zinc or have too low levels of copper present, then the enzyme can't work at all. And so here you can see in red is the cofactor, and it's actually making this enzyme, this blue thing, work so that the substrate can come in and bind to the active site. If this red cofactor wasn't here, the enzyme wouldn't be able to work. Next, it asks us to describe the effects of heavy metals or poisons on enzymatic activity. Just remember that just like high temperature and extreme pH, heavy metals at high concentrations can cause enzymes to denature. And what that means again is enzymes are proteins that have a very complex three-dimensional folding to them. When they denature, they lose all that folding, and therefore they lose their active site. The substrate can no longer bind to it. Objective 5 says interpret graphs of the effects of pH and temperature on rates of enzymatic reactions. So as I said before, the relationship you always see between the rate of an enzymatic reaction and temperature is that it will increase to a certain point and then decrease because of denaturation. However, what I want to point out here is that every enzyme has a different optimum temperature. So in this case, you can see that enzymes typically found in our body, and our body temperature is typically around 37 degrees Celsius. So enzymes in our body would show a trend like the red line here. The optimum temperature for their performance would be around 37 degrees Celsius. However, enzymes that might be found in maybe a fish living in the Arctic, where it's really cold, might have an optimum temperature around 4, where they'll function the best. However, there are some bacteria that live in hot springs, which temperatures in hot springs are near boiling, or maybe are boiling. And in this case, those enzymes would have a very high optimum temperature. They have to be able to function really, really well in very, very hot temperatures. So every enzyme has a different optimum temperature, and that all depends on where they're found. If you look at pH, these are 3 enzymes found in your body. Pepsin is found in your stomach, and notice it has an optimum pH around 3, and that's because your stomach is very acidic. So the lower the pH, the more acidic that means. A pH of 7 is very neutral. Amylase is found in your saliva. Your saliva is very neutral, has a neutral pH, so that's why amylase works best in that environment. Arginase is an enzyme found in your urea cycle. It helps to produce your urine. Your urine is very basic, so arginase, this enzyme, functions best, has the fastest rate of its reaction, and a pH that's around 11 or 12. So here we have a practice exam question, and this is a more difficult question, so if you can answer a question like this, then you should feel pretty good. So it says, based on the graphs, what is most likely true about the enzyme urease? Well, if we look at this graph is showing the effect of pH on urease activity. This graph is showing the effect of temperature on urease activity. Well, let's look at each one of these questions now, or at each one of these answers. So the first one says, urease is denatured at a pH of 5.5. Is this true? Well, it doesn't look like it. It looks like it's pretty active. Urease activity is really high at that pH. So I'm going to cross this one off so I know that that is not correct. Its optimal temperature is 67 degrees. Yeah, well, it looks like that's optimal. That's where the activity is the highest. So let me circle this one. Let me just make sure the other two are not correct. Its activity increases as temperature decreases below 55 degrees. Well, as temperature decreases below 55 degrees, the activity is decreasing, not increasing. So yep, that's definitely wrong. It functions best in a basic environment, so pH greater than 7. Well, definitely not. It's really low there. So yep, B is the only one that is correct. Objective 6 says, describe the effects of substrate and enzyme concentration on rates of enzymatic reactions. So one thing you want to know is that if we increase the substrate concentration, but the enzyme is limited, then we're going to see a relationship like this. The rate's going to increase but then flatten out. If the enzyme wasn't limited, if we had excess enzyme, then the rate of the reaction would continue to increase. But since we're talking about a situation here where the enzyme concentration is limited, the rate's going to flatten out. So let's look at why this happens. So as you increase substrate concentration and the enzyme is limited, the rate will level off. And if you look here at this example, each test tube has the same amount of enzyme. So the enzyme is the limiting factor here. But we're going to increase the substrate in each test tube and see how it changes the rate. So in order for a reaction to happen, the substrate and the enzyme have to come together. So if we look at the beginning and we have a small amount of substrate here, we can see that in our example, we can have one reaction happening at a time. So our rate would be 1 reaction at a time. If we increase our amount of substrate even more, we can see now we can increase the rate. We can have 2 reactions happening at a time. Let me increase it even more. Well, now it's 3 reactions at a time, so I've increased the rate. But let's see what happens if I increase the substrate even more. So I increased it now to 5 substrate, but you can see because the enzymes are limited, how many reactions can happen at once? Only 3. So my rate levels off. If I increase the substrate concentration even more, my rate is still 3 reactions at a time. So my rate eventually will level off, and that happens because all the active sites are filled. In other words, the active sites are saturated with substrate at a certain point. And at that point is when the reaction will actually start leveling off. Right here, once all the active sites are filled, the reaction rate levels off. Let's see what happens when you increase enzyme concentration. Well, you're going to see a similar effect. If the substrate concentration wasn't limited, you would see that it would continue increasing and increasing and increasing. But if the substrate concentration is limited, then we see a plateau as well here. So here we see that our enzyme concentration is going to be increased, but we have a limited amount of substrate in each test tube. So let's see what happens to the rate. Here, 1 reaction can happen at a time. Let's add some more enzyme. 3 reactions can happen at once. Add some more enzyme. 5 reactions can happen at once. But now, even if I increase and add more enzymes, what's going to happen to my rate? Well, the maximum rate that could be reached is still 5 reactions at a time. If I increase my amount of enzymes even more, the maximum rate is still 5 reactions at a time. In this case, there's not enough substrate, so the maximum rate eventually levels off. Objective number 7 says describe the enzymatic activity in terms of the lock and key model and relate this model to the effects of temperature, pH, and heavy metals. So what this is saying is that the substrate and the enzyme are very similar to a lock and a key. We know that only 1 key fits in 1 lock, and it's similar that only 1 substrate can fit with 1 type of enzyme. So 1 type of substrate and 1 type of enzyme, and they fit together perfectly like a lock and key. So this all has to do with the active site. The active site of an enzyme has a specific shape to it, and this shape will only fit that of 1 type of substrate. Even if another reactant came along, so another molecule wanted to be changed, and it tries to fit into this active site, it's not going to fit, and it will not be changed. Only the substrate can be changed by an enzyme. So, as I said, only 1 key can fit 1 lock, or 1 type of key can fit 1 type of lock. Only 1 type of substrate can fit 1 type of enzyme. So how does this relate to the effects of temperature, pH, and heavy metals? Well, temperature, pH, and heavy metals alter the shape of this active site. So if I change the shape of this active site, will my substrate be able to be changed? Will my substrate fit anymore? No, it will not. So denaturation, which can be caused by high temperatures, extreme pH, or heavy metals, will mess up the lock and the key. Okay, well, specifically, it will mess up the lock, so the key can't fit in it anymore. So here I have a couple really important properties of enzymes that you should definitely know for the exam. Know that enzymes are usually proteins. This means they have a very complex 3-dimensional shape. If you alter that shape at all, the enzyme will not work. Enzymes are very specific, so they only work for 1 type of substrate. Enzymes are unchanged in a chemical reaction. So however many enzymes you start with, that's however many enzymes you're going to end with. They don't increase or decrease in the reaction. An enzyme's shape is crucial to its function, as I stated. So denaturation and things like that can really affect an enzyme. If an enzyme loses its shape, it will no longer be able to change anything, to interact with the substrate. And enzymes have an optimum temperature and an optimum pH where they work the best, and this all depends on where they're typically found and the type of organism they're typically found in. Furthermore, you want to know factors that can affect enzyme performance. We know that temperature can affect the performance of an enzyme, that as temperature increases, the rate will increase to a certain point, and then the rate will decrease. And that is because of denaturation that it starts to decrease again. You see a similar trend with pH as well as enzymes. Let me draw a graph, x-axis and y-axis. Just as in temperature with pH, it goes up and then down. In these extreme pHs, denaturation is occurring. Heavy metals will also affect an enzyme. High concentrations of heavy metals will decrease the rate of a reaction. The presence of a cofactor will affect an enzyme as well. If the cofactor is present, the enzyme will work. If the cofactor is not present, then the enzyme cannot work. Remember, cofactors are things like iron or lead or other things like that that typically are metals that need to be present in order for an enzyme to work. And enzyme and substrate concentration will also affect the rate of a reaction. If either the enzyme or the substrate concentration is limited, then the rate of the reaction will most likely go up and then plateau as you increase enzyme or substrate concentration. So here I have a couple examples. Let's test your knowledge on enzymes. Which statement is false about enzymes? First says they are catalysts. Well, catalysts are things that cause change but are not change themselves. And enzymes are catalysts. So they're the same thing. So this is true. Enzymes, they lower the activation energy of a reaction. That is true. They lower the amount of energy that's needed to activate the reaction. They are the key in the lock and key model. Well, actually, enzymes are the lock in the lock and key model because enzymes are the ones with the active site. So this is false. The last one says they are highly specialized. That is true. So C is the correct answer here. What is the term for a molecule that is required by an enzyme in order for it to function properly and is also unchanged in a chemical reaction? Well, if it's required for an enzyme to work, a catalyst is an enzyme, so that's not the answer. A substrate is also required for an enzyme to work, but a substrate's changed in a chemical reaction. The product is the result of a reaction, so the answer here would be a cofactor. The cofactor is a thing that needs to be there, but it also is not changed in a reaction. An enzyme and a cofactor are not changed. All the following causes an enzyme to denature except, well, we've already talked about this. Extreme pH, yes. High concentration of substrate, no, that's not going to really affect the shape of an enzyme at all. So that's not going to do anything. Heavy metals would cause denaturation, and high temperatures would also cause denaturation to happen. All right, let's look at another example. Lactase breaks down lactose into glucose and galactose. Lactose intolerance occurs when the small intestine does not make enough lactase. In this example, glucose is So a lot of students freak out when they see a question like this, but just break it down. So lactase breaks down lactose. So lactase is causing the change, right? So lactase is the enzyme. So let me write that up here. It's causing the change. It's the one breaking it down. It's breaking down lactose into glucose and galactose. So what is the thing changing? Well, lactose is the thing that is changing. So we know the substrate changes. So this is the substrate. Well, what's it being changed into? It's being changed into glucose and galactose. So it's asking what is glucose? Well, glucose and galactose are the result of this reaction, so they are the products. Also, one thing I want to point out is that enzymes tend to end with ACE. So that's one tip to help you realize which molecule is the enzyme. So every time you read a question like this, ask yourself, okay, where's the enzyme? What's causing a change? What is being changed? That's the substrate. And what are the results? Those are the products. Here's another question. This one can always trick students up. A student wants to know the optimum temperature for the enzyme zymase. This is our enzyme. He did a floating disk experiment and his results are shown in the table below. So this is showing the number of disks that rise per second. So the higher the number, the faster the rate, the more the reaction's happening. What is the optimum temperature? Well, this is pretty simple. It's 40 degrees. That's where the rate is the highest. If the student says the rate at 37 degrees was 0.065, so 37 would fall in between here, would he be interpolating or extrapolating? Oh, which would he be doing? Well, since it's within the range of data, he has some evidence to back that up, so it would be an interpolation. Okay, here's another question. Doctors become concerned when a patient has a very high fever because enzymes in the brain can become malformed. What is the term to describe this change in shape of enzymes? You better know this word. It's a really important one, and that's denaturation. So that's a result of high temperatures usually. So I want to point out this question here. On the exam, don't forget that if a statement is false, that you also correct it. Okay, so that it's true. If you don't correct the statement, you won't get credit for that problem. So here it says a catalyst is a molecule or mineral ion that is required by an enzyme in order for it to function properly. Well, catalyst is not the correct word here, so that's false. You cross out catalyst, and what should go there? Cofactor. So you would just cross that out and write cofactor on top. I think that's the last question. Yep, it is. All right, so that's Unit 4. If you have any questions, just let me know.